The Keynote 427 study is a study of frontline pembrolizumab in untreated renal cell carcinoma patients. It has two cohorts. It has a clear cell cohort A and a non-clear cell cohort B population. And the study presented at ASCO this year is looking at that study and using tissue samples to do a biomarker analyses. The clinical endpoints for that study have previously been reported. The cohort A population of 110 clear cell patients has been shown to have an overall response rate of 37%. And so we're looking at that patient population with an RNA-seq-based assay and defining coherent gene signatures, multi-gene expression profiles that include a T-cell inflammation profile and 10 other consensus gene signatures, and looking for an association with overall response rates in this cohort. And the findings were that the T-cell inflammation gene signature did associate with overall response rate, but none of the other 10 consensus signatures. There was not a strong association with other clinical endpoints, progression-free survival or overall survival. So I think, again, this study, 110 patients, it is a smaller study than some of the large frontline phase 3 trials, but there has been an effort to do biomarker analysis across many of the frontline clinical studies for renal cell carcinoma. And our data does correspond to observations that have been made from the combination of atezolizumab, an anti-PD-L1 antibody, plus bevacizumab, and observations made from the Javelin study of avelumab plus axitinib, that T-cell inflammation at baseline does seem to associate with superior clinical outcomes. Now, that isn't true across all of analyses that performed in the same way. At ASCO this year, there was also biomarker data presented from the Checkmate 214 study, that's frontline ipilimumab, nivolumab. So pure immunotherapy, if you will, in the frontline setting, did not show a strong association with any of the biomarker analyses that were tested in that cohort. There was also a very large data set of PD-1 blockade monotherapy from pooled studies, primarily from previously treated patients receiving PD-1 monotherapy, that again had limited ability to define clear-cut biomarker observations. There is a T-cell subset within that overall analysis that does seem to associate, again, with improved clinical outcomes. So the theme of T-cell inflammation at baseline within tumors that may predict a better overall survival, better overall outcome, whether it's overall response, whether it's progression-free or overall survival, depending on how the study has been designed. But what to do with that information? Is this really poised to change our clinical practice? And I think at this point, the answer is no. The patients that do not have the biomarker still generally fare rather well with our checkpoint blockade. So although it's interesting and we can enrich for the response phenotype, it's not a discrete enough separation of the population that it's going to be a marker that is going to be used as a diagnostic to include patients for checkpoint-based therapy. So I think the field is still in search of more discrete or more powerful markers that might better select patients that are going to have the uniquely good outcomes, or select patients that are destined to have primary refractory disease where we might move in a different direction with their primary therapy. So although I think interesting in providing some context about how the drugs work, I think not yet ready to impact our routine clinical management.